Okay, so here's quiz 1.6, and topic 1.6 focuses primarily on nucleic acids. So understanding the structure and function of the nucleotides and the differences between the two major kinds of nucleic acids that we are going to learn about in this class, DNA and RNA. The first diagram here we see, a nucleic acid fragment that is made up of 4 nucleotides linked together in a chain. It asks which of the following characteristics best shows that the fragment is RNA and not DNA. So it actually, there's, unless you happen to have memorized, which you do not need to, it is well beyond the scope, the chemical signature of the different acid groups here, the A's, T's, C's, G's, and U's, you wouldn't be able to tell because it doesn't label the different nucleotides here. So you can see that it has 5' to 3' orientation. That's true. But both DNA and RNA have that. So we cannot tell that this is RNA and not DNA from that characteristic. The identity of each nitrogenous base, if we knew the identity of each of these bases, then we would be able to tell the difference between them. That would show the difference. The charges on the phosphate groups are the same for all of them. And then the types of bonds that link the nucleotides together, again, it's the same for all of them. So these 3 things, A, C, and D, are the same for DNA and RNA, so that wouldn't help us differentiate them. The only possible correct answer here is that it would be the identity of the nitrogenous base. Sorry for my cough. It's just a cold, by the way. I found out yesterday that I do not have the COVID. Well, late last night. DNA and RNA are nucleic acids that can store biological information based on the sequence of their nucleotide monomers. We have a short segment of each of the 2 types of nucleic acids. Which of the following best describes the structural difference between those? Now, this one is a tricky one. Most of you selected the correct one. The backbone contains deoxyribose of DNA, whereas RNA contains ribose. And you should know that. You should know the main structural differences between DNA and RNA. And one of them is the sugar that's in the backbone of the DNA. Again, you're going to just need to be able to draw out a DNA molecule. You should be able to label the phosphates, the sugars, the different bases, and you should be able to tell me the differences between the bases in DNA and RNA. You should be able to tell me how many hydrogen bonds form between AT groups and CG groups. There's a different number. So CG bases form 3 hydrogen bonds, ATs form 2. That makes ATs easier to unzip. That's going to come up later on in the year, so this is a repeating theme. And you also need to remember the difference between the 5' end and the 3' end of the DNA molecule. I'll show you that in just a second, because that's kind of a tricky idea, one which I might have gone over in class with you yesterday had I been there. The one people selected that was incorrect is that a DNA molecule is composed of 2 parallel strands with the same 5' to 3' directionality, whereas RNA is composed of only 1. This is one where you needed to read it carefully to figure out that this one is wrong. When you first read this, you're like, yep, 2 strands in DNA and 1 strand in RNA. And that's true. And if that's all they had said, you would not have been able to select between B and C. They both would have been correct. However, they do say that the DNA strands are parallel with the same 5' to 3' directionality, and they're not. DNA, the 2 strands are not considered parallel. They're considered to run anti-parallel. So DNA has a direction that it travels. It is not just a line traveling in 2 directions continuously. It's traveling in 1 direction. That directionality helps us differentiate the 2 strands because one strand is running up and the other strand is running down. Now, as far as that 5' to 3' directionality, I think The last diagram will show that for us. No, the first diagram will show that for us. So they explain this in the video, but here's the basic idea. Here's your sugar, your ribose or your deoxyribose, depending upon whether it's DNA or RNA. The way you figure out where the 5' is when we count, or when we look at a hexagonal shaped sugar molecule like this is always true, whether you're looking at glucose or ribose or whatever, galactose. Each of these corners is a carbon atom. And that's just the way organic chemists draw them. So at each of these bends is a carbon atom. So there's a double bonded oxygen here, an oxygen bonded to two different carbon atoms that sort of folds the carbon up into this shape. And the way you count carbons is you start at the oxygen and you go clockwise. So this corner is carbon 1, carbon 2, carbon 3, carbon 4. And then you go up and there's another little bend right here. So that's carbon 5. So this is considered the 5' end of the molecule because it's the 5' carbon that's free and ready to bond. But if we look down here on the other end, the 5' carbon is already connected to a phosphate group which is bonded to another nucleotide. So instead here we go 1, 2, 3, 4, 5. But it's the third carbon, this OH right here, that is free to bond to another nucleotide. So if another nucleotide came in, it would bond to this group right here. It would knock off the hydrogen. It would be a dehydration reaction. It would take the OH that's normally bonded to the phosphate off. That would form water. And you'd end up with a phosphorus oxygen bond here to bond the next one. So there's a 5' end and a 3' end. And that's important to remember. You need to be able to identify that and know what it means. Because later on we're going to trace the direction of how DNA is read and put together and things like that by tracing the direction of the 5' to 3'. In the last question, we have these two diagrams of DNA and RNA. We've got the cytosines and the adenines and stuff labeled, and it says, which of the following conclusions most clearly supported by the representation of nucleic acid number 1 and number 2? And everybody got this one right. So it doesn't seem that there was any problems with this one. This one is simply testing, do you know that nucleic acids, that DNA contains AT base pairs and CGs, and whereas RNA, any base pairing that happened would happen between, it doesn't even contain thymine, it contains uracil, so there would be no T's in the first place. And also there isn't bonding between base pairs in RNA because it's just single nucleotides. So there we go. Please let me know if you have any questions.